The next thing I want to talk about as it relates to prokaryotic cells is a structure that we sometimes find in those cells that's unique to prokaryotes. And this structure is called a cellular inclusion body. So it's important to note right away that an inclusion body is not an organelle, and that's because it's not surrounded by a piece of membrane. These are little compartments inside of some prokaryotic cells. You don't find them in all prokaryotic cells. But I like to think of them as little storage units because that's what they're used for. They're used to hold on to certain materials for later usage, basically. They're actually found in a lot of different species of bacteria and archaea. And you can see inclusion bodies underneath the microscope. Now, what do we find in these little storage units? Some inclusion bodies are actually there to hold on to lipid compounds. Remember, the lipids include fats. And just like in eukaryotic organisms like us, fats are a great way to store energy for the long term. So some prokaryotes put lipid into inclusion bodies as a way to store fat, just in case they need it at a later date. It's long-term energy storage for them. There are other inclusion bodies that hold on to small carbon-containing molecules that the cell might need later on to synthesize things like protein and carbohydrate and so on. Carbon reserves, we refer to these as. There are other inclusion bodies that store minerals. And finally, there are what I think are the most fascinating inclusion bodies, and those are the ones that hold magnetosomes. On this slide, you can see some prokaryotic cells under the electron microscope. So this is one cell right here. This is a rod-shaped cell. And inside this cell, we can see these large white structures. These are not organelles. These are inclusion bodies, and they happen to be full of lipid. So you can almost think of this as a big, fat prokaryotic cell. It is just loaded with lipid inside. So it will hold on to that lipid. It will use that lipid as energy storage for later on. For later on, it can burn the fat if it ever gets into a situation where it doesn't have enough energy. It doesn't have enough nutrients in order to provide energy to itself, so it will burn the fat for that reason. Down here on the lower part of the slide, you can see several rod-shaped cells. And inside several of these, there are, again, these big white sort of cavities. These are lipid-containing inclusion bodies for energy storage. Now, these cells are actually starting to break the lipid down. You can see that you don't see a nice, big, perfect inclusion body here. Instead, the white is sort of breaking apart, and that's because these cells are breaking down that lipid. They're in an environment right now in the culture where they don't have enough nutrients for energy. So they're going to use their fat stores for energy. Now, I mentioned the magnetosome, and I said it's the most interesting type of inclusion body, at least I think so. These are inclusion bodies that contain literally magnets. So the magnetosome, if you translate that term, it means magnet body. So these are inclusion bodies that have magnetic material in them. And in this image, this is a single prokaryotic cell that has several of these magnetosomes in it. Now, what a cell does with these internal magnets is it orients itself in a magnetic field. If you think about it, a prokaryotic cell needs to know where it is in its environment. It doesn't have eyes or ears, it doesn't have the senses that you and I have, but it still needs to know where it is. Where is, north and south and east and west? And the magnetosomes help it do that. They will orient inside the cell according to whatever magnetic field that cell is in, and that will align the cell along certain directions. Very, very interesting structure. Now, the last structure I want us to talk about today is called an endospore. And you may have heard of spores before. An endospore is a spore that forms inside a prokaryotic cell. Now, there are other types of spores. For example, the fungus can make spores and release the spores into the environment. Endospores are different. Endospores form inside prokaryotic cells, so inside bacteria and archaea. And what an endospore is basically an inanimate form of that prokaryotic cell. It is literally a way for that prokaryotic cell to put itself into suspended animation. Now, only a limited number of prokaryotic cells can do this. Essentially, this process involves the living, metabolizing form of the cell, what we would call the vegetative stage of the cell, going through a process called sporulation and creating these inanimate structures called endospores. The endospore then is a dormant structure. It is an inanimate structure. It's not metabolizing, it's not dividing, it's not doing any of the things that we would associate with a living cell. It's not really alive at that moment, but it's capable of coming back to life. So spores are really fascinating. You can see spores under a light microscope because they are very refractive. They bend light in a certain way, and so they have a particular look. They almost look mirrored underneath the microscope. They sort of flash underneath the microscope. Now, there are also special stains for endospores that help us see them clearly under the microscope. So what would cause a prokaryotic cell to sporulate? What would make it go from the vegetative sort of metabolizing, dividing stage of life into this inanimate spore? This only happens under very certain circumstances. The number one thing that will drive a cell into sporulation is drying, desiccation. As a cell begins to dry out, that is a moment that brings that cell close to its death. And that will trigger that cell to sporulate if it's able to. The other thing that will do it is poor nutrient availability. If the cell finds itself in an environment where there is just not enough nutrients, that will trigger that cell to sporulate if it can. So spores are survival structures for prokaryotic organisms. They are inanimate. They are not really alive in that moment. They sort of put that cell into suspended animation, and they hold its DNA, it's that critical genetic material. They hold that DNA in this suspended animation until the environment improves, until there's more water around and there's more nutrients around. Now, what makes spores particularly fascinating is that they are very resistant to destruction. So if you put them in under high heat, for example, or you expose them to harsh chemical disinfectants, or you even expose them to types of radiation, they don't get destroyed. So they're very resistant to destruction. Now that becomes important in medicine because the last thing we want to do is have spores on, for example, our surgical instruments and things like that. In order to destroy endospores, you have to either autoclave the material or you have to apply certain disinfectant chemicals for an extended period of time. And if you're not familiar with autoclaving, we're going to talk about it in detail later in the summer, but for now, understand that it's a process that applies both heat and pressure to materials to sterilize them. What you're looking at on this slide is a drawing of this process called sporulation, as well as a couple of images of cells that are going through the process. So start up at the top of the slide where we're looking at just some generic prokaryotic cell. Maybe this is a bacterium and it's a rod and it's in an environment right now where either there's not enough water or there's not enough food. There aren't enough nutrients for it. So it's going to start sporulating. What's happening inside the cell during this moment is it sort of swells up, and they've shown us that by showing the cell getting longer and longer. And it's developing this structure inside of it called, again, an endospore. Once the spore is completely formed, the rest of the cell just breaks away and disintegrates. The most important parts of the cell, though, the DNA especially, is now inside of this mature endospore. Now, when the conditions get better, this endospore can regenerate into a vegetative cell, and that's what they're showing us here. We call that process germination. Once there's enough water and enough nutrients, that spore can go back and become a normal, metabolizing, dividing vegetative cell again. Remember, spores are very resistant, and because they're inanimate, because they are not metabolizing, they are not dividing, they're just sort of suspended in the moment, they can last for long, long periods of time. So on the right-hand side of this slide, up at the top, we can see some bacterial cells in culture, rod-shaped cells again. Now we're going to stress these cells. We're going to take away a lot of the water, or we're going to take away a lot of the nutrients, and this starts to happen. Now, you can see that a lot of these cells have started to sporulate. I know that because there are these circular structures inside some of these cells. And this is what I mean when I say you can see these under the microscope. They are very sort of perfectly spherical, and they have almost a mirror-like appearance. You can't really see the mirror shine unless you're looking under the microscope, but trust me, you can see it. Right over here, and right here, you can see two spores that have lost the rest of the cell. These are mature endospores. They have finished the process of sporulation. On this slide, we're looking at a smear of bacterial cells. Again, these happen to be rods, and they have been stained with a very special kind of stain that we use to see endospores. As you see on a lot of these cells, not quite all of them, but a lot of them. Most of the cell, most of the rod-shaped cell is colored red, but then there's this structure inside that's colored green. The green structure is the spore. And yes, there are a couple of spots on the slide where the spores are sort of released. You don't see the red cell anymore. Those are mature endospores. Here's another picture taken under a light microscope. This is a rod-shaped bacterium called Bacillus subtilis. This bacteria is sporulating. Doesn't have enough of something. It either doesn't have enough water or it doesn't have enough nutrients. And as you can see, in some of these cells, there is a sort of refractive white colored structure. And in some of the areas, it looks like it's surrounded by pink. That is the endospore that's developing inside the cell. Most prokaryotic cells that are capable of forming spores are actually found in soil. Most of the species of bacteria and archaea that are capable of sporulating are actually in the soil. And so they're not harmful to us. It makes sense that if you live out in the soil, in the world, you're going to encounter times when your environment gets very dry or you run out of nutrients, right? Depending on the time of the year. So it makes sense that a lot of soil organisms can sporulate. Pathogenic spore formers, the kinds that can cause disease in us, are limited. There are only 2 genera. And remember the word genus, as we've talked about, the scientific name, the part of the name that is the genus. If you use that word as a plural, it's genera. So there are 2 genuses or 2 genera of bacteria pathogenic bacteria that can form spores. Those are Bacillus and Clostridium. So remember those 2 names. Any organism whose name starts with Bacillus or whose name starts with Clostridium are spore formers, any pathogenic organism, I should say. For example, there's an organism named Bacillus anthracis, and this is a spore-forming type of bacteria that causes the disease called anthrax. There's a bacterium called Clostridium tetani, which is the bacterium that causes tetanus. Both of those are pathogenic bacteria that are capable of forming spores. Endospores are very resistant to destruction, and they can persist in the environment for a long time. In fact, we don't know how long spores can last and still come back to life. We think, at least so far, that they can last for thousands of years. It's amazing. We have found, for example, spores inside the wrappings of Egyptian mummies. And when we have taken those spores into the laboratory and put them into a culture with plenty of water and plenty of nutrients, they have reverted back to vegetative cells. So spores that are thousands of years old can still revert to living cells once they have the things they need to grow. So when it comes to pathogenic bacteria like the Bacillus and the Clostridium, we have to think about them in medicine, because if we accidentally placed those endospores, those pathogenic endospores, into a human body, what we have done is we've given the spores a warm and wet and nutrient-rich place, right? The human body is very warm and nice and wet, and it's loaded with nutrients, and those spores will germinate and go back to become vegetative cells and cause disease. So we have to be very careful when we are sterilizing medical equipment that we do it correctly to make sure that we destroy any pathogenic spores that might be on that equipment. This last slide I put in just to show one of these pathogenic spore formers. This is an image up at the top taken with a scanning electron microscope. These are anthrax spores. Anthrax spores are problematic not just for humans, but also for other types of animals, especially farm animals, large animals that are out in the environment. Down at the bottom here, you're looking at a person's arm. Their elbow would be up here, their hand is over here, and this poor person has anthrax. This person has a form of anthrax called cutaneous anthrax. So the spores got onto this person's skin and got the moisture they needed and the nutrients they needed to germinate and go back to the metabolic vegetative form of the cells, and then they created these horrible cutaneous ulcers. Anthrax has a very characteristic look. We refer to these as eschars, these black sort of scabby looking regions. This is very characteristic of anthrax. Now, we don't see a lot of anthrax under normal conditions, but occasionally cases pop up. We have reason to fear that anthrax could be used as a biological weapon. If somebody were able to take these spores in a large enough number and disperse them, for example, with a bomb or some other device, you could get a lot of people very ill very quickly. So our government actually vaccinates our military members for anthrax, just in case someone were to use a bioweapon of anthrax against us. And there have been a couple of cases in our history where people have tried to send anthrax spores through the mail and have successfully done it. People who are capable of growing the bacterium up in the laboratory and then stressing it out so that it sporulates. These terribly mentally ill individuals have actually spread anthrax through the mail from time to time. So it's a terrible disease. The cutaneous form of anthrax is actually quite treatable with antibiotics. But if you inhale anthrax spores, you develop a form of the disease that is the respiratory form. And as you might imagine, that form of anthrax is usually quite fatal. Very interesting organism caused by Bacillus anthracis.